Okay, go ahead.
Yeah. Hello, everyone. I welcome Dr. Prasanna for our third webinar of Genome India International. And it's a great pleasure for all of us to have Dr. Prasanna in this webinar. So welcome you, sir. And I also welcome all the participants. And it's really great to have you today. So before going to the webinar, I would like to give a brief introduction about the GII. And this was initiated in the year 2005 with the aim of networking and sharing the knowledge in the field of genomics and to help the younger generation and to motivate them. And it has been a wonderful experience in last several years. But recently we have started this webinar with the leading experts in this field. So in the Genome India, this international was the lead founding president, Professor Kole is the founder. And there are several members in the committees who are taking lead in different areas. Today, Professor Kole could not join and he has apologized to the speaker and also to all the audience. So with this, I invite Professor Prasanna, and he is the director of the CIMMYT Global Maize Program, and he has a very vast experience in maize genetics, genomics, and breeding. He has been focusing on developing maize varieties for resistant to disease, insect pest, and improving the nutritional components. He has also extensively published papers, more than 140 publications he has made in this field, and several more than 50 book chapters, and also received several awards in this field. He is a member of National Academy of Science and also other academies in this field. So with this brief introduction, I welcome Dr. Prasanna and ask him to deliver the talk. Thank you, sir.
Thank you very much, Genome India International, for giving me this opportunity to provide a perspective on genomics-assisted breeding for maize improvement based on the work that CIMMYT does together with partners, especially in Sub-Saharan Africa, Asia, and Latin America. I'll particularly focus on genomics-assisted breeding, although there is a vast amount of work that also done on various other aspects of maize improvement at CIMMYT. But especially for this audience, I would focus on this. But before we go into that, this is an excellent publication from FAO in 2021, highlighting the state of food security and nutrition in the world. And they highlight in this volume 4 major factors that are influencing are challenging the food and nutritional security. One is climate variability and extremes. The second is conflicts around the world for various reasons, including the recent conflict between Russia and Ukraine, the economic slowdowns and downturns that have been happening due to various factors, including transboundary diseases and pests affecting human and animal and plant health, and the unaffordability of healthy diets due to volatile prices of the major foods. So if you look at this, plant breeding has a major role to play, especially in tackling the various variabilities and extremes that come due to the changing climates, as well as other devastating aspects like pests and diseases. South Asia and Sub-Saharan Africa are indeed hotspots for climate-induced variabilities and extremes. If you look at it in Sub-Saharan Africa, the size of this circle indicates the quantum, the number of people, millions of people that are affected on average due to climatic variabilities and extremes. So the Horn of Africa, which we call this, especially in Eastern Africa, is highly vulnerable to climate-induced variabilities. Similarly, publications from the Asian Development Bank clearly show that South Asia is especially highly vulnerable. You can look at the high sensitivity as well as the low capacity to withstand such climatic variabilities is highly prevalent in case of South Asian countries compared to East Asia, like for example, in China. So both these make our task much more complex in terms of how to tackle these climatic variabilities and extremes through multi-pronged strategies. Maize is a major crop around the world. Almost more than 180 million hectares of maize are presently grown globally. Of this, approximately 70 million hectares are under the rain-fed, stress-prone environments in sub-Saharan Africa, Latin America, and Asia. That's what is the target of CIMMYT. These approximately 70 million hectares of rain-fed maize, not irrigated maize. And these rain-fed maize is highly vulnerable to an array of abiotic stresses, including drought, heat, waterlogging, diseases, as well as insect pests, the most recent of which is the fall armyworm. Yet, this is the region where maize provides a major contribution to the total calorie intake, almost 15% on average in South Asia, to over 50% in case of several countries in sub-Saharan Africa and Latin America, where maize is a major staple food. So increasing crop yields in such stress-prone tropics cannot come from one particular discipline alone. We require genetic innovations on one hand, very strong seed systems on the other hand, and improved agronomic management practices. So a combination of solutions along with enabling policies are required to sustainably increase crop yields in general, including maize, and the livelihoods of smallholders who depend on crops like rice, wheat, maize, grain, legumes, roots, tubers, and bananas. So where do the impacts of CIMMYT's maize program, global maize program, stem from? First of all, very strong regional collaborative maize breeding and seed systems networks, including not only the national partners, but private sector all around the world. The second is the germplasm coupled with extensive phenotyping and testing network in the tropics. If you look at it, we host managed stress phenotyping trials on our germplasm at multiple hubs across Latin America. For example, you look at Central America, Mexico, Colombia, and in Africa, in Kenya, Ethiopia, Malawi, South Africa, Zimbabwe, and if you look at South Asia, India, Bangladesh, Nepal, and Pakistan. These are our main major breeding hubs, and all these are hosted by the national partners as well as the private sector partners. In addition, our germplasm reaches multiple other countries where they are evaluated outside these major phenotyping hubs. Then strategic partnerships on modern maize breeding and professionally managed breeding and phenotyping platforms, offering shared services to national programs as well as small and medium enterprise seed companies. Breeding must always start with the demand-driven product profile. It's a good understanding of where can you focus, where do you need to focus, what is your comparative advantage in that particular market segment where you have where you can offer much more than what any other competitor can offer. So this is where we prefer to focus on specific product profiles in Eastern Africa, in Southern Africa, in South Asia, and in Latin America. These are multiple stress-tolerant maize product profiles combining trait combinations involving abiotic and biotic stresses. In addition, we also have niche markets for nutritionally enriched maize, especially the provitamin A enriched maize with more than 33 ppm, more than 15 ppm provitamin A, or high zinc with more than 33 ppm kernel zinc. So maize program of CIMMYT, together with partners, focuses on product profiles, defined product profiles for specific market segments, combining multiple stress tolerance and nutritionally enriched traits. You cannot be successful, genomics assisted breeding cannot be successful without having a very strong grip on phenotyping, and especially for the traits of your interest. So that's what CIMMYT's main strength is, to have high heritabilities for the trials that involve measurements or evaluation of our germplasm for drought stress. For example, this is a picture taken from Kibako in Kenya, or heat stress at Hyderabad, where we are hosted by ICRISAT, or waterlogging stress. So, and providing clear differences in terms of not only the vegetative stage response, but also in terms of yields. You can look at how a heat-tolerant maize hybrid from CIMMYT performs under high vapor pressure deficit stress compared to a commercial benchmark check in the same environment. Or similarly for drought stress, the drought-tolerant hybrid of these versus susceptible hybrids from the private sector, or the waterlogging stress. So precise phenotyping is fundamental for improving any breeding program. Maize is no exception to that. The second most important aspect is the breeding programs then need to focus on what are the key elements to improve the genetic gains in the various breeding pipelines, focused around these four important elements: selection intensity, selection accuracy, functional genetic variation, and reduction of cycle time, which is the denominator here, and intensity and accuracy. Whether it is with regard to the phenotyping network, for example, this is our heat stress phenotyping network across South Asia, and clearly defined on the basis of vapor pressure deficit, high vapor pressure deficit, moderate vapor pressure deficit, and low vapor pressure deficit sites. The second important assisted breeding, genomic selection. These contribute selection accuracy. We need to have excellent managed stress phenotyping protocols, harmonized protocols, coupled with opportunities to reduce or account for the field variability wherever such managed stress screens are being implemented. Functional genetic variation can come from the understanding of which particular gene bank accessions have complementary or highly favorable haplotypes that can enrich your traits, as well as modern day tools like genome editing can also improve your functional genetic variation. This is our genome editing efforts right now for MLN resistance implemented in partnership with Kotava. You can see here a highly resistant line coupled with a highly susceptible one, side by side, evaluated at Naivasha, which is our MLN or maize lethal necrosis dedicated screening facility in Kenya. In terms of reduction of cycle time, again, there are multiple opportunities. You can implement 3-season nurseries per year using seed dryers. And that's what we do at Kibako in Zimbabwe and so on, to be able to implement very fast breeding cycles. Secondly, double haploid technology. I will come to that a bit later, or speed breeding done in some other crops. We don't have to do speed breeding in case of maize because DH is a very powerful technology available in case of maize. So you can reduce the cycle time again through multiple avenues. The next most important thing, all this is relevant if you have annual and routine monitoring of genetic gains in breeding pipelines. I'm sorry to say that a number of breeding programs cutting across crops, cutting across different types of organizations have no credible means to or credible efforts to actually evaluate genetic gains in their breeding pipelines. This is a major limitation in many countries. And this needs to be rectified very well, not only through what we call era studies that compare different varieties that have been released in different eras, but also to be able to analyze historical data to evaluate the genetic trends in your breeding pipelines. And third important thing, annual and routine monitoring of genetic gains by comparing your pipeline materials in comparison with the commercial benchmark checks as well as internal genetic gain checks. So breeding pipelines must annually monitor, and this is a high-level key performance indicator. This, through various statistical means, you can then compare if you have good check connectivity over years. Your test entries could differ from year to year, but because of this overlapping commercial checks or internal genetic gain checks, the statisticians or the quantitative geneticists can use those as anchors to evaluate your genetic gains. You also need to have a check replacement strategy, not replacing all checks in one go, but overlapping checks. Target population of environment coverage. So evolved breeding pipelines or breeding programs have systematic approach to improve genetic gain per dollar invested. And that's the stage at which we would like to see most crop breeding programs go. And this is a publication. We have been annually monitoring genetic gains. That's a different aspect. We also done era studies, but in November 2022, we came up with this comprehensive publication, which is a first global study to estimate genetic trends across CIMMYT's tropical maize breeding pipelines in Eastern and Southern Africa, South Asia, and Latin America over an 11-year period, with analysis of data from 4,152 advanced breeding trials conducted at over 1,300 locations in 28 countries globally, showing a very impressive genetic trends for grain yield reaching up to 130. So we are second to none in terms of improving genetic gains, even when we are working under highly stress-prone environments in Sub-Saharan Africa, Asia, and Latin America. The second aspect is reducing the breeding cycle time. As I mentioned, the double haploidy is a powerful tool in the maize breeders' armory to do that. What we did since 2007, we embarked upon a tropicalized haploid inducer development strategy. And by 2012, we released the first generation haploid inducers, natural haploid inducers through systematic selections and going up to 10% haploid induction rate. By 2017, we increased it to now over 10 to 13% haploid induction rate in the tropical genetic backgrounds. And not just developing it and using it on our own breeding pipelines, but we have disseminated globally. Over 30 public sector institutions and over 32 seed companies from 24 countries have sourced CIMMYT's tropicalized haploid inducers since 2012. So that is the first most important thing. We wanted to make much wider use of haploid induction and DH development process in the tropical environments and not just limited to multinationals. Then we established centralized platforms in Kenya at Kibako, in Agua Fria in Mexico, and most recently at Kunigal in India, in partnership with US Bangalore, a double haploid facility. These centralized platforms offer maize DH development service not only to CIMMYT breeders, but also to the national partners and to the small and medium enterprise seed companies in the respective continents. Over 80% of all inbred lines used or parental lines used in our CIMMYT maize breeding pipelines globally are now doubled haploid lines. So we can increase significantly genetic gains by using DH in combination with breeder-ready markers for specific traits, as well as through rapid cycle genomic selection. Then the power of DH increases exponentially if you use DH in combination with breeder-ready markers and rapid cycle genomic selection. So this is an excellent avenue for shortening the time required for recycling elite lines in the maize breeding pipelines. When it comes to the genomics-assisted maize breeding, this is a summary. In essence, how do we do that? When the number of haplotypes are more than 1 to 3 for a given trait, then you have to go for genomic selection because the number of markers required for integration through more forward breeding or through marker-assisted backcrossing are too many. So in that case, we can conveniently go for genomic selection or genomic prediction. But if it is limited to just one haplotype or maximum 3 haplotypes, we can go for marker-assisted forward breeding for trait enrichment in our breeding pipelines, marker-assisted backcrossing if you have to convert a susceptible but elite line into a resistant line, or marker-assisted recurrent selection to accumulate the frequency of favorable alleles in your populations. So if you look at it in different breeding pipelines in Eastern and Southern Africa, we use different strategies: genomic selection or genomic prediction for improving grain yield under drought or under optimal conditions, and for key traits like maize streak virus, maize lethal necrosis, we go for marker-assisted forward breeding or backcrossing. For some of the traits where there are more than 3 or 4 haplotypes, we go for genomic prediction, like for example, flowering time or GLS or TLB resistance, and so on. So this is where you need to have a clear understanding of the genomic architecture for a specific trait and adapt a suitable strategy in terms of using breeder-ready markers or genomic selection to improve the trait in your breeding population. Why trait markers? In terms of, for example, why do you need to discover breeder-ready markers for some of the traits? This provides an assayable variation that can be tracked in successive generation, because these markers are closely associated or sometimes within a gene that confers a trait of interest. So they are typically faster. It is possible to select a seed or seedling stage without waiting for what we call grain yield, or without waiting for flowering behavior to happen. Selection can be done in trait insensitive environments. For example, I can do selection for MSV resistance and MLN resistance in Latin America without having those traits phenotyped in that particular continent. Permit the selection and enrichment of populations with heterozygous individuals through codominance. It is substantially cheaper in high throughput, low density setup, and you can also pyramid precisely high value trait loci, like what we do, for example, provitamin A enrichment plus maize streak virus resistance plus MLN resistance using markers itself in some of our breeding pipelines. But before you embark upon such a trait discovery, marker discovery project, you should ask some critical question. Is there a demand for the trait in the context of the product profiles targeted? How efficient and what is the cost of existing methods of selection in the breeding process? If it is too high, the phenotyping cost is very high and the efficiency is not so good, then if there are markers, it's certainly marker-assisted breeding is is the choice strategy to embark on. The possibility of improvement of various factors contributing to genetic gain through incorporation of marker-based selection at the early stages in the breeding scheme, and the genetic diversity available for the trait in the breeding pool and the need for rapid trait enrichment to achieve your product profile deliverables. So these 4 questions are extremely important before we say that, yes, I need to try and embark upon experiments or trials to discover marker or genomic regions that contribute to a particular trait of interest. And at CIMMYT, we have a clear stage gate advancement process. This is a marker trait pipeline. So discovery project proposal comes to me, for example, Prasanna, I would like to do a discovery of genomic regions for Falamiwam. Yes, then you go through the 4 important traits. Then, okay, this is really important trait for us to embark upon a discovery project. Then the breeder, the molecular breeder, together with the respective phenotyping team and the breeding team, goes ahead with the discovery of this, either through GWAS or through the linkage mapping, if you had to validate in independent populations what you discovered through genome-wide association studies. Then this is from this stage to the next stage, whether we go for validation in breeding populations or technical validation of genotyping assays, the molecular breeder needs to submit data. And that data will be critically reviewed by our team before it goes into a stage gate advancement into the next stage. And then once this is done, in terms of validation in breeding lines or on technical validation of genotyping assays, then the third stage happens. That means, are you ready for deploying those markers either in marker-assisted forward breeding or backcrossing or for recurrent selection? So if you look at this, different traits are at different stages. Some are already under deployment, some are under technical validation, and some are under discovery and validation in independent populations. So an evolved crop breeding program needs to have not just a random approach towards marker trait pipeline, but systematic progression from one stage to another stage of marker trait pipeline. So once this is done, look at the MSV resistance. We routinely sample all the lines at a very early stage in Eastern and Southern Africa for the presence or absence of a specific haplotype. This is a haplotype on chromosome 1 with a very high contribution to phenotypic variance. And then you select lines that are favorable with the favorable haplotype and reject those lines which do not have that favorable haplotype. And that's how, much before you do even test crossing, you can reject a number of double haploid lines which are segregating for the favorable and unfavorable haplotypes. This is a typical marker-assisted forward breeding approach. And this is much more powerful rather than doing simply marker-assisted backcrossing into some elite lines. That means here we are making markers or breeder-ready markers a routine or an integral part of your breeding scheme. So we use this not just only for one particular continent or one particular breeding hub. We can see here how we are introgressing favorable alleles for targeted traits from breeding pipelines in Latin America and South Asia. That means exchange of germplasm. You don't have to evaluate these traits only after they go to Africa. You can do this marker-based analysis even when you do these experiments or early stages of your breeding work in Latin America for certain traits, in South Asia for certain traits. That's how you can see lowland tropical Latin American lines crossed with Africa, or CIMMYT germplasm resource line crossed with Africa, an XPVP line from US crossed with Africa adopted line, or a South Asia line crossed with an Africa adopted line. So this is an important part of your breeding strategy. How to exchange lines between breeding hubs and how to use breeder-ready markers because you are evaluating in trait-insensitive environments. We also did marker-assisted backcrossing. For example, when MLN has become a major problem in Africa, in Eastern Africa, we discovered very quickly a highly favorable QTL on chromosome 6 in one of the lines, and then validated in independent populations, devised the technical assays for that, and quickly transferred this favorable allele from that source materials into multiple drought-tolerant elite lines that are used as parental lines in several of our commercial hybrids. As many as 52 such elite lines have been converted from MLN susceptible lines into MLN resistant versions just within a matter of 2.5 years. That's the power of, again, discovery of breeder-ready markers and quickly mobilizing them into your elite lines and converting them into from susceptible to resistant versions. So this is one of the major achievements. Imagine 52 lines converted, parental lines converted into resistant versions. So in terms of stage gate, we have moved in the 1C GIR. It's not just CIMMYT. So together with IITA, we do this work across a range of traits. For example, this is an example of our October and November stage gate advancement meetings together with molecular breeders, not only from CIMMYT, but also from IITA. So this is a 1CGR Maize marker trait pipeline, and the decision whether we require more data on validation, whether we can go into further refinement in deployment strategy, whether we can go into validation in forward breeding populations, or we can directly go into deployment, or sometimes if you are not satisfied with this, explore other selection strategy. Your data is not good enough for this particular trait. So that's the kind of an approach you need to take in terms of systematization of this entire decision-making process. Now I'll move into genomic prediction or prediction accuracy. How good is good enough? So when we started this work, much ahead of the crop breeding programs that we know in the CGIR system, our goal was to approach the realized heritability of the current stage 1 testing in our breeding pipelines and ask the question, can we predict the performance of a large number of untested lines in the coming season with roughly the same accuracy as if we had 4 to 5 environments of phenotypic data on each tester? It's a tough question. So, but this is where evolved breeding programs need to reach, not just publishing papers on genome sequencing, and but actually move into routine integration of genomics prediction in your breeding pipelines. Phased implementation of genomic selection is what we gone into. In phase 1, we used some full SIPs to reduce the overall cost and build an across-year training set for a given heterotic group into breeding pipeline. So that means we expanded the population size significantly. In phase 2, we used half SIB and other relatives from the previous years to sample more G by E year, genotype by year data, and eliminate radically 1 year of testing altogether, and thus saving the cycle time significantly and also the phenotypic burden and saving the cost to the breeding programs. How did this translate in terms of results? So if you look at it, genomic prediction sufficiently is accurate to replace 50% of our stage 1 testing effort. So we developed models, prediction models, with 50% DH in stage 1 testing. That means genotype all the DH lines, but phenotype only 50% of them and predict the other 50% based on genomic estimated breeding values. Based on this, we advanced top 20% of our double haploids to stage 2 testing. And then in phase 2, we replaced even 100% of our stage 1 testing effort by training multiple years historical data. I hope colleagues are following this. So this is a radical shift in thinking in terms of understanding genomic relationships, estimating genome estimated breeding values, reducing drastically your phenotypic burden, and moving very rapidly in terms of your breeding cycle. That's how we moved from year 2015, when we started on phase 1, to year 2020 onwards into phase 2. So in 5 years, we made the transition. And we showed through various publications how genomic selection and phenotypic selection are quite comparable. That means there is no loss of accuracy while reducing significantly the time required. So genomic selection, initial approach we followed was test half and predict half in 4 different pipelines for grain yield under optimum, drought, low nitrogen striga, saving up to 35% of our resources. Then from test half, predict half strategy to test quarter and predict 3/4 strategy by data. Sources up to 52% with the same selection accuracy or efficiency as phenotypic selection. So genomic selection is a very powerful tool. If you use it in combination with the sparse testing, so in a breeding program like Maize, we use typically 3 to 5 testers. Earlier, each line used to be crossed with each of the 5 testers, and it used to have a tremendous phenotyping resources required for this. Now we don't even have to cross every line with every testers. You can use the sparse testers, and you can even use sparse locations in terms of experimental designs, and that can save resources a lot. So this is called rapid cycle genomic selection. We started in EAPP1, one of our product pipelines. We moved to 3 product profiles in Eastern and Southern Africa with 3 cycles per year of marker-based selection and updating of our historical database training populations. The cost benefit analysis, we have done that. Phenotypic selection, testing in stage 1, genomic selection making test process, phenotyping training set in stage 1, genotyping all lines. So the total cost of genomic selection is 91,870 in a particular pipeline at stage 1, compared to a total cost of almost $135,000. So you are saving almost $45,000 on each breeding pipeline. That is the genotypic selection by phenotypic selection cost ratio. So 32% saving straight away in your breeding cost. So historical data, as I said, can replace your early stage of testing, stage 1 sometimes. And this is what we published, Tanda and the team in 2021, how time and cost efficiency can be done by using multiple biparental populations in your training set and selected using algorithms to maximize the relatedness between the training set and the tester set or prediction sets. For populations with close relatives in the historical data sets, phenotyping lines at the stage 1 yield trends could be completely eliminated. Using only then GBVs or genomic estimated breeding values to advance the new lines into multi-tester, multi-location yield trials. So this entire strategy we summarized in this publication in terms of it's like a decision-making tree. What do you have? You test half, predict half. How do we combine with sparse testing? How do we use algorithms to select the most informative relatives to augment your full SIP data? And how to use the historical data to train your populations. So this strategy, if we need to put the pieces together, forward breeding and the genomic selections are quite complementary. So you can use them together with the double haploid based strategy in terms of putting together again a decision tree. If there is a large effect haplotype segregating, go for forward breeding. Use it in combination with the double haploids, and then you can go for, and if it is also amenable for GCA genomic selection, use genomic selection and straight away go into stage 2. But if there is, the population is not amenable for GCA based genomic selection, then go for stage 1 and stage 2. So this is the conventional inbreeding strategy has been completely now replaced with the DH based and genomic selection and marker assisted forward breeding. In essence, friends, genomic selection is not an add-on to your conventional breeding. It needs to be implemented as an integral part of your breeding process and your breeding pipeline. It has to align with your breeding goals and strategies, and it needs to be a part of your broader efforts to improve the efficiency, both in terms of cost and the time taken for your breeding strategy to be implemented. So you need a well-oiled machinery, a team with a clear understanding of their roles. You need capacity development and shared services in terms of genotyping and implementation of foundational capabilities and well-defined what we call standard operating procedures. And always consider phased implementation strategy like what we did. Test half, predict half. Then we move into historical data and then eliminate even one stage of testing altogether. Now, from forward breeding and backcrossing to genomic selection, there is a third most important application of molecular markers, that is quality assessment and quality control throughout your breeding and seed value chain. So in case of maize, we have a defined set of markers, and we published a manual, Manjagowda and team, a few years back on how to use a defined set of SNPs, and we have well optimized assays to evaluate the genetic purity and identity of the lines that you are using in your breeding program, as well as the parental lines of hybrids that are released by partners. So this evaluation is systematically undertaken, not only in our own breeding pipelines, but also we also provide technical backstopping to the national partners and seed companies so that they can ascertain the purity and identity of their materials throughout their seed value chain. The final aspect is about on-farm testing. It's not only on-station testing that has given us the impacts, but we have stage 1 to stage 4 is on-station testing. Stage 4 is a regional on-farm test, on-station testing with multiple partners across our target market segments. But stage 5 is a selected set of stage 4 hybrids that go into extensive regional on-farm testing. And these on-farm testing is done completely under farmer management conditions. So we include appropriate commercial checks, farmers' local varieties, and 3 or 4 of our excellent stage 4 hybrids. And in an incomplete block design, we evaluate them over hundreds of locations across Eastern and Southern Africa. This is called modified tricot approach. So from approximately 100 on-farm testing sites 2 years back, we have expanded this to over 700 testing sites in Eastern and Southern Africa. 40% of them are women managed. We GIS map each of these locations in that particular market segment. We have online digital data capture and central repository where the partners can easily provide the data. They don't have to send information by data capture on books and sending it to us. Digital capture of the data, which automatically goes into our centralized repository, and this has improved our data turnover as well as efficiency. So through this extensive regional on-farm testing, we used to evaluate and we continue to evaluate the stage 5 hybrids, which are at the end of the product development spectrum. Once the on-farm data is collected, then we again do product advancement meeting, taking the on-station and on-farm data and selecting which amongst these products should be actually announced to partners through CIMMYT website. But what are we gaining from this on-farm network? A better understanding of G by E by M, genotype by environment, by management, by the farmers, environment in the target population environments. Decipher in-depth the differences of the gender-related varietal and trait preferences and use them in our final product advancement. Incorporate that more robust data on farmers' preferences in selection of lines for recycling, as well as products, elite products that will be advanced and announced to partners. And provide feedback to the seed companies as well as policymakers, because we are testing our test entries versus the best commercial checks in that particular market. So what we have recently embarked upon is a major change from that. Not only we are doing now a stage 5 end of the spectrum product advancement using on-farm. We are now evaluating our stage 1 materials. That means these are at the early stages of breeding scheme, also on-farm, on-station, as well as on-farm simultaneously. So this project is called Maize on-farm genomic selection project. So where we are going to generate genomic estimated breeding values, not only based upon our on-station performance data, but also taking the on-farm data. So it's a very challenging project. We are piloting this in 2 breeding pipelines. The first CG breeding program to actually start doing this at the early stages of breeding, on-farm preferences are taken into consideration right at the early stages of breeding schematic. All this will be effective once you have a very effective product licensing models. And we do this not just breeding, but also product licensing to both national partners and private sector partners. So since 2017, our diverse product licensing models have enabled 275 unique maize hybrids and 11 improved open pollinated varieties to be licensed to 197 distinct partners, including 37 national partners, 4 NGOs, 6 parastatals, and look at this 150 seed companies. Many of them are small, medium, as well as large seed companies. For registration and commercialization across 70 countries globally. That's the impact, the geographical imprint of our products going to diverse countries across the world. And what is the impact of this is the next question, not just varietal registrations are released, Certified seed production of our CIMMYT-related varieties, improved maize varieties, has in fact tripled between 2016 to 2021, from almost 72,000 tons to 170,000 tons. This is overall CGIR, including CIMMYT plus IITA, out of which 130,000 tons roughly are from CIMMYT genetics. So that means from less than 1 million hectares in 2010, in 2021, approximately 5.25 million hectares are reached with CIMMYT-related improved germplasm, benefiting estimated 32 million people only in Eastern and Southern Africa. Simultaneously, the area-weighted average age of varieties reduced from 14 to 15 years in 2015 to less than 10 years in Southern Africa and just 10 years in Eastern Africa. And overall across Eastern and Southern Africa, 10 years. This is a huge improvement in terms of area-weighted average age of CIMMYT-related varieties in Eastern and Southern Africa. So my final slide. So breeding programs and seed systems need to continuously evolve. Efficiency, collaborative network, and culture are extremely important. In terms of organization, we need to look at the changes in the external world that are driving the need for change. In terms of technologies, we need to look at what are those disruptive innovations that need to be integrated faster in our crop breeding programs. In terms of structure, what are the changes in the operating practices to become more and more efficient? And in terms of process, what physical assets, capabilities, and roles need to change if we have to move the entire breeding process in a much more efficient manner. It's not a job of one individual. It's the job of the entire team to coordinate and to collaborate more effectively and bring such changes in a very rapid manner. And behind all this work, there are a lot of partners. For example, in Sub-Saharan Africa, Asia, Latin America, the CGIR initiatives, the funders, including Bill and Melinda Gates Foundation, USAID, FFAR, they are all key partners in this whole venture. Thank you so much.
Thank you very much, Dr. Prasanna, for a very interesting and highly informative talk. And you have really focused on hello, can you mute the speaker? Yeah.
Yes, sir, I'm muted.
So it was a very interesting talk, and you have really highlighted all the points, starting from germplasm evaluation, phenotyping, and for the traits like drought, waterlogging, all these traits are very difficult to phenotype. And this is very important, accurate phenotyping, then collecting the data, sharing the data, and finally also highlighted about the genomic selection, the importance of double haploid, and how you can make use of these technologies for developing new varieties. And what we have seen is that due to climate change, new insect diseases are coming up, and again, stress for different abiotic stress is also increasing. So this is a very interesting talk, and I'm very confident that this technology will be very helpful to the farmers in all over the countries, different countries, and to increase the food production and in the maize breeding program. So once again, thank you very much, Dr. Prasanna, and we will now start the questions answer program. And if there are any questions.
So I hope people can unmute and ask questions if possible.
Yeah, I request participants that please feel free to ask the questions, because it is very important to interact and to gain knowledge with this expertise. Yeah, Dr. Prasanna, I would like to ask just some basic questions, like what motivated you to initiate research in this area? So we would like to know, and how exciting and was the journey
Yeah, I
for?
think, yeah, of course, I have been in maize breeding since my PhD days. So I have stuck to maize for over 30 years. And even after 30 years, I know only very little about maize. A lot still needs to be learned about maize. That's how powerful the crop is in terms of its amenability to genetics and breeding. That said, of course, we started off through Asian Maize Biotechnology Network in India, the first lab to focus on marker-assisted evaluation of genetic diversity, not only in elite lines, but we did this in land races collected in India as well as worldwide through the Generation Challenge project. Then we moved into marker-assisted breeding for traits like quality protein maize, downy mildew resistance, and so on. But when I joined CIMMYT in 2010, it provided an opportunity to systematically elevate the game of CIMMYT in terms of double haploid, in terms of marker-assisted breeding, genomic selection, high throughput phenotyping. So I didn't cover many other aspects that made us very impactful. And also a seed systems team. We have a dedicated seed systems team that interacts with the private sector and ensures that the varieties that we have developed and released to the partners actually go to the farming communities. Ultimately, whatever we are doing to the breeding pipelines, the real impact is when those products actually reach the farming communities and at a large scale. That's the ultimate success of any breeding program. You can assess genetic gains and all that, and you can show impressive progress. But if your final products are not competitive enough, then your impact will be very limited. So it's not just the publications on genomics or genomics-assisted breeding, but very practical focus on how to integrate markers in your breeding strategy, how to reduce the cost, and how to improve the efficiency, and how to use multiple technologies in combination to drastically increase genetic gains and to derive impactful products. Whether it is maize, rice, legumes, or any crop or wheat, this should be the mantra in terms of using a practical-oriented genomics-assisted breeding strategy.
Yes, yes, please go ahead, Dr. Manish.
So, Sajid, just have one question. So, like in genomic selection, right now, we are factoring in, like, say, genotypic data, phenotyping data, environment data. And where do you see that the role of, like, say, soil nutrition and then maybe microbiome, whether these should be also the integral part of those, like, say, as an factor when we are making the decision? What is your take on this?
Yes, initially, to me, I think we need to move in a systematic, phased manner, understanding the genomic relation matrix within the lines that you are using and reducing the phenotypic burden through what I showed, gene test half, predict half strategy, or test quarter, predict 3/4 strategy. That's the phased implementation. Then you can enlarge it upon by taking the environmental variables, like what we are going to do in the maize on-farm genomic selection project is to also take into consideration the farmer typologies, including what you are mentioning. There are farmers. This is like 1,000 farms. And out of these 1,000 farms, we form different clusters these are relatively homogeneous units of farmers who adopt certain practices. These are all, remember, under farmer management conditions. Some will apply certain levels of fertilizers. Some will use intercropping. Some will have a different soil status. So there are different practices adopted by the farmers. Together with the socioeconomics team and the environmental GIS team, you can cluster these diverse or heterogeneous farmers on farm, which are randomly sampled into clusters of homogeneous units. And those clusters could be used as a covariables when together with your on-station and on-farm performance data in terms of understanding the genomic estimated breeding values. Remember, in this case, the selection unit is not a line per se. The selection unit is actually the haplotype, different haplotypes that are present in different breeding lines. That haplotype could be present in multiple lines that are in stage 1 testing. So that's the difference, Manish. It's not the line per se, which is the selection unit here, but the haplotypes, understanding those haplotypes and which haplotypes are getting evaluated in which farmers, and how do these farmers rate the products, and then you go into the haplotype selection. So you can use the environment and management variables into as a co-variables when you analyze the genomic estimated breeding values.
Thank you,
I
sir.
hope
Thank
this
you.
answers your question, Manish.
Yeah, very much.
It's not so easy. You do need to have excellent socioeconomic input,
Yes.
excellent understanding of your target population of environments, and strong biometrical support.
Any other questions from the participants?
So I often joke that I served as a faculty member of IRA genetics division for almost 2 decades, teaching several courses there. So when students are silent, either they understood completely or they didn't understand anything. Okay, I hope it is the former, not the latter.
It was a very interesting, and as we have seen in several crops, to come up to this level, it is not easy to apply all these technologies, and including genomic selection and then developing varieties. And what you have pointed out is varieties are being developed, but are they reaching to the farmers? That is a very key point. And seed, sometimes good quality seed is not available to the farmers. So that point also you addressed. And also global partnership with so many countries is the key. And really, we are very happy to see this program, and we would like to have your expertise in other crops also, so that speed breeding and developing new varieties can be made.
Sure.
So with this, once
Absolutely.
again, yeah, once
Thank
again,
you
I
so
think
much. Thanks for this opportunity, and please feel free. Any of you, if you would like to ask any question, my email ID is there. b.m.prasanna. Somebody asking a question in the chat box.
Yeah.
I would like to know how functional genomics, cis regulatory sequences, CRISPR help us more to understand or to use genomic selection. Yeah, if those functional genetic variation is clearly linked to a trait of interest. That means they do contribute significantly to the phenotypic variance. Then you can use them as a part of your either, for example, marker-assisted breeding strategy. So when you look at the marker trait pipeline, if some of those genomic regions have clear value, it need in terms of contributing to phenotypic variation, then you can, and if they are limited in number, as I said, not more than 1 to 3 haplotypes, use it in marker-assisted forward breeding. But if you want to use them in genomic prediction, you can also use a mixed model where some of these genomic regions could be used together with a random set of markers in your prediction strategy. So both ways it would be helpful. CRISPR is, of course, a completely different strategy. Genome editing undoubtedly would require well-defined gene of interest. That means when you understand, for example, I will give you an example of MLN resistance. When we started off, we did genome-wide association study, identified a major region on chromosome 6 that contributes to a significant proportion of phenotypic variance, validated in independent population. Then we fine-mapped the genomic region, and then we went to the gene of interest together with Kotava. So characterizing the gene of interest enabled us to understand what polymorphisms within that particular gene could actually lead to MLN resistance. And then those became the targets for us to undertake the CRISPR-Cas9 strategy in terms of getting genome-edited MLN-resistant lines. So your marker-assisted breeding strategy can provide you with very vital leads to go to fine mapping and to identify the gene of your interest. These are not mutually exclusive. Remember, if you want to do CRISPR-Cas9 based improvement, you do need to have a very firm understanding of what gene you are targeting and what are the polymorphisms within that particular gene that lead to your trait conversion, right? So that's where you have a very effective complementarity between your marker trait pipeline and your genome editing approach. Okay, how do you connect a large heterogeneous group into homogeneous covariable group? On what basis? That's what I said. You need to cluster the farmers based upon specific traits, specific things that are, for example, management practices that are common. Like, for example, as I said, in Africa, there are different types of typologies of farmers. Some farmers use no fertilizer at all. Some farmers use 20 kg nitrogen per hectare. Some may use 40 kg nitrogen per hectare. Some areas get affected by certain types of stresses more. Some areas get certain other kind of stresses more within a particular target population of environments. So you need to take a detailed database look into what farmers are, how farmers are managing their particular farms. We have a detailed questionnaire, and before we go on an on-farm data evaluation network, we get detailed understanding of the farmer typologies. And we don't select only those farmers who are closer to the road or who are what we call doing certain practices that are more optimum. You need to go into more interior regions and sample those smallholder farmers who are not often reached by improved innovations. So selecting that group of farmers who come to hundreds of farmers, remember 700 to 1,000 farmers in a particular region, then we do it together with the socioeconomics team and the partners. Map them systematically, GIS, and understand their typology. And then use the data variables. And before that, cluster into relatively homogeneous clusters. Okay, that's good. So I'm successful today. At least there were 2 questions from students or research scholars. Okay, so I can have my dinner, no problem.
It was a very interesting, and we would be in touch with you for any further queries regarding this area. And once again, I thank Dr. Prasanna, Dr. Manish Pandey, Dr. Manit, and all the participants. It has been very nice to see everyone here. And we will again meet in our next webinar. And so once again, thank you very much. Thanks to all.
Thank you so much. Thanks a lot, Genome India, especially Milinji and Manish and colleagues for organizing this and giving me an opportunity to interact with all of you. Thank you so much. Bye-bye.
Thank you, sir.
Thank you, sir. Thank you.
Bye, everyone.